The functional unit in the nervous system is a neuron. Neurons receive and send the neural impulses that trigger an organism's response to the environment. A neuron consists of a cell body, dendrites, and an axon. The cell body contains the nucleus and all the organelles found in the cytoplasm. Dendrites are short extensions of the cell body that receive stimuli. The axon is a long, slender extension that transmits an impulse from the cell body to another neuron or to an organ. The cone-shaped region of an axon where it joins the cell body is called the axon hillock, where the signals that travel down the axon are generated. Each branched end of an axon transmits information to another cell at a junction called a synapse. At most synapses, chemical messengers called neurotransmitters pass information from the transmitting neuron or presynaptic cell to the receiving cell or postsynaptic cell. Neurons are not always transmitting signals. The membrane potential of a resting neuron is known as the resting potential. Potassium ions and sodium ions play critical roles in the formation of the resting potential. In humans, the concentration of sodium ions is higher in the extracellular fluid than the concentration of potassium ions. The reverse is true inside the axonal membrane, where the concentration of potassium ions is higher in the cytosol than the concentration of sodium ions. The sodium and potassium ion gradients are maintained by sodium-potassium pumps in the plasma membrane, which use the energy of ATP hydrolysis to actively pump 2 potassium ions in for every 3 sodium ions it pumps out of the cell, which leads to a net loss of positive charges within the cell. The concentration gradients of the potassium and sodium ions across the membrane represent a chemical form of potential energy. Converting the chemical potential to an electrical potential involves selectively permeable ion channels. The ions carry units of electrical charge as they diffuse through channels. The net movement of positive or negative charges will generate a voltage or potential across the membrane. In a resting neuron, there are many potassium channels open, but very few sodium channels that are open. Since the concentration of potassium ions is much higher inside the cell, the gradient favors a net outflow of potassium ions, which leads to an excess of negative charge inside the cell. The resting membrane potential is always negative inside the cell, and the neuronal membrane is said to be polarized. In humans, the negative charge is negative 70 millivolts.
Action potential is a massive change in membrane voltage. It results from positive sodium ions depolarizing the membrane. The action potential will occur whenever a depolarization increases the membrane voltage to a certain value, which is called the threshold.
Resting potential is the membrane potential characteristic of a non-conducting excitable cell, with the inside of the cell more negative than the outside.
The threshold is the potential that an excitable cell membrane must reach for an action potential to be initiated. Depolarization is the reduction in the magnitude of the membrane potential. It usually involves gated sodium channels. Repolarization is the change in membrane potential that turns it to a negative value just after the depolarization phase of an action potential. Hyperpolarization is an increase in the magnitude of membrane potential, and in general, it results from any stimulus that increases either the outflow of positive ions or the inflow of negative ions. The refractory period is the short time immediately after an action potential in which the neuron cannot respond to another stimulus, owing to the activation of voltage-gated sodium channels. This creates a maximum frequency of action potentials. Saltatory conduction is the rapid transmission of a nerve impulse along an axon, resulting from the action potential jumping from one node of the Ranvier to another, skipping the myelin-sheathed regions of membrane. The first step in creating an action potential is the resting state. Here, the gated ion channels are closed. The second step is depolarization, in which a stimulus opens some sodium channels. Positive sodium ions flow into the cell. If this depolarization reaches the threshold, it will cause an action potential to occur. The third step is the rising phase. Most sodium channels are opened, and most potassium channels remain closed. This causes the inside of the membrane to become positive. The fourth step is the falling phase. Most sodium channels are inactivated, whilst most potassium channels open. Positive potassium ions flow outwards, which makes the inside of the cell negative again. The final step is the undershoot. The sodium channels close, but some potassium channels are still open. The membrane will return to its resting state once these potassium channels close and the sodium channels become unblocked. Synaptic vesicles are membranous sacs containing neurotransmitter molecules at the tip of a presynaptic axon.
A neurotransmitter is a molecule that is released from the synaptic terminal of a neuron at a chemical synapse, diffuses across the synaptic cleft, and binds to a postsynaptic cell, triggering a response. In a chemical synapse, the postsynaptic membrane is the membrane that receives a signal from the presynaptic cell and responds via depolarization or hyperpolarization. The postsynaptic membrane is separated from the presynaptic membrane by the synaptic cleft.
One of the most common neurotransmitters in both vertebrate and invertebrates is acetylcholine. Most neurons that form a synapse with muscle cells release acetylcholine as an excitatory transmitter. It binds to receptors on ligand-gated channels in the muscle cell, producing an EPSP. Acetylcholine activity is terminated by acetylcholinesterase, which is an enzyme in the synaptic cleft that hydrolyzes the neurotransmitter. In regulating vertebrate cardiac muscle, acetylcholine has inhibitory effects. It activates a signal transduction pathway. The G proteins in the pathway inhibit adenylyl cyclase and open potassium channels in the muscle cell membrane. This reduces the rate at which cardiac muscle cells contract. Some amino acids also serve as neurotransmitters. Gamma-aminobutyric acid, or GABA, appears to be the neurotransmitter at most inhibitory synapses in the brain, and it produces IPSPs by increasing the permeability of the postsynaptic membrane to chlorine ions. Glutamate is an excitatory transmitter in the brain. Glycine acts as an inhibitory transmitter in the central nervous system outside of the brain. Biogenic amines are neurotransmitters derived from amino acids. Norepinephrine acts both as neurotransmitters and hormones. It has both excitatory and inhibitory effects. Dopamine is excitatory, but may be inhibitory at some sites. Serotonin is synthesized from tryptophan and is generally inhibitory. Neuropeptides are short chains of amino acids that serve as neurotransmitters, and they operate via signal transduction pathways. Substance P is an excitatory neurotransmitter that mediates our perception of pain. Endorphins are inhibitory and serve as analgesics that decrease the perception of pain. Some gases also serve as neurotransmitters. Nitric oxide can be excitatory or inhibitory and produced in the peripheral nervous system.